So rabies is arguably the most deadly viral disease known to humans. Upon the onset of symptoms, it has a near 100% case fatality rate. And despite the existence of an effective vaccine, tens of thousands of people still die of rabies every year, mostly in parts of the world with inadequate healthcare resources. So a small molecule therapeutic that demonstrates efficacy during the symptomatic stage of the disease would have a huge impact on this very ancient and still very significant public health need. So Prosetta, where I work, has been developing a highly unconventional approach to small molecule drug discovery, which is being applied to numerous viral families. But today, I'm going to talk specifically about the application of this approach to the discovery of a compound with potent activity against rabies. So our platform is predicated on two major hypotheses. The first one is that viral capsid assembly occurs via a host-catalyzed biochemical pathway involving discrete assembly intermediates and energy-dependent steps, in contrast to the more conventional view that capsid assembly is a spontaneous thermodynamically driven process. Our pathway-based model presents catalytic host-virus protein-protein interactions that can be thought of as previously unappreciated opportunities for antiviral intervention. The second big hypothesis is that the host factors involved in this pathway are components of very heterogeneous, labile, and dynamic multi-protein complexes, which are not amenable to conventional purification techniques, and therefore not amenable to specific target protein-focused drug discovery programs. So then how do we pursue these drug targets? And we start with a classical cell biological technique, cell-free protein synthesis, where we take a cytosolic extract, supplement it with amino acids and an energy regenerating system, and program it with mRNA transcript for the protein that we want to express. And using this, we've expressed rabies capsid proteins. This gel shows an audio radiogram of 35S radiolabeled rabies nucleoprotein, matrix protein, and phosphoprotein, and all three co-expressed in the cell-free system. And what we can do is take translation products from this system and analyze them for rate sedimentation on sucrose step gradients, and thereby separate out different points, steps along this pathway. So we adapted the cell-free system into a drug screen, basically in the form of a modified ELISA. And what it is really is just a fluorescent plate assay that monitors for the formation of multimeric rabies nucleoprotein containing complexes. And it allows us to screen for small molecules that interfere with this pathway at any of numerous steps with absolutely no knowledge of what their potential targets might be. So the readout that we get from this screen looks like this. And here I show an inactive compound compared to an active compound where you have a dose-dependent diminution in fluorescence. And the other thing we do in this screen is we co-express EGFP, which is completely inert on assembly, but allows us to identify false positives that are diminution in signal just based on diminution in, or inhibition of protein synthesis. So you can see in this hit that I'm showing here that that's not the case. So hits identified on the screen are then subjected to SAR optimization, and here's a series of analogs to the active compound from the previous slide, and their activity on the cell-free screen. And we corroborate this activity with activity against infectious virus in cell culture. And what you can see is the extent of activity corresponds well enough to validate our platform and our working hypothesis for this approach to drug discovery. So we picked one of these compounds, PAV866, to move forward with for further study. And here's its structure. So if our hypothesis that these compounds are acting on catalytic interactions, their efficacy in cell culture should be entirely dependent on their addition within a very specific time frame, right? Basically in time for protein synthesis and assembly. So we did an experiment where we added the compound at varying times in the first 12 hours of a 72-hour experiment. And so if the compound was acting directly on the progeny virus, any time within those time points shouldn't make a difference, because there's still plenty of time for the compound to act on the virus once it's released in the medium. But what we see is that anytime after 6 hours post-infection, the compound loses activity. And this is totally consistent with compound action on an intracellular target within our proposed pathway. So then to retroactively identify the target, we took advantage of the high affinity of PAV866 to the target and coupled the compound to a resin, basically to use it as a ligand for affinity chromatography. And we applied the cytosolic extract that we use for the cell-free protein synthesis reactions to a column of this resin. And what we found was a set of proteins that specifically bound and then eluted with the free compound. What we show here is 2 different cellular extracts, total extract, and then a control column of blocked resin, and then the PAV866 column, where you can see the set of proteins that bind. And we took the flow-through from this column, basically the cytosolic extract depleted of the proteins that are binding to our compound, and put that on our cell-free plate screen. And what you can see is the nice dose-dependent titration from the starting extract is completely lost in the depleted extract. But when the depleted extract is reconstituted with the eluates from the compound column, exhaustively dialyzed to remove any free drug, you see a full restoration of the compound effect. So this is showing that these proteins binding to the column do in fact represent the functional target of our compound. To further characterize this target, we took the starting extracts and we ran them on glycerol gradients, and found that these proteins largely migrate together, peaking in fractions 3 and 4 of the glycerol gradient. And what we're seeing is sort of starting to see a picture of this assembly machine that is comprised of multiple protein subunits associated together in this complex. We've been identifying these proteins by mass spec, and I'll talk about one of them specifically, which is ABCE1, which is a host factor that's previously been implicated in HIV capsid assembly. And it gives us a window into sort of the mind-boggling heterogeneity that we're dealing with here. This Western blot, the top panel shows total starting cellular extract applied to a glycerol gradient and then Western blotted for ABCE1. And you can see that it's very heterogeneous. You've got ABCE1 in every single fraction of the gradient in multiple different forms. And when these fractions are applied to the compound columns, only a tiny subset, a very specific subset, binds to the column in fraction 3 there. And notably, that is a different subset than is associated with HIV capsid assembly. So what we're seeing here is one protein, ABCE1, that's involved in potentially lots of different of these multi-protein complex machines. And so targeting this kind of drug target, if we were to approach it with a technique like siRNA knockdown of ABCE1, we would never be able to identify this drug target because we would not have the selectivity we would need to target this tiny subset of the protein. A little more about mechanism that we're starting to look at is programming our cell-free translation reactions with truncated mRNA rather than the full-length mRNA. This is just missing the stop codon so that a large number of the nascent chains are retained on the ribosome. And when we put those on sucrose gradients, you can see the first panel there, the polysomes with the nascent chain attached, and then we release it with puromycin, and they migrate to the top of the gradient and then progress through the assembly pathway as they would normally. And when we put these truncated mRNA reactions on the plate screen, we can dissect the timing of the drug effect based on compound addition relative to puromycin release. When we add the compound prior to puro release, we get the drug effect. And when we release with puromycin before adding the compound, we lose the drug effect. So this implies a very early mechanism, as early as even while the nascent chain is still attached to the ribosome. Although we've shown with EGFP that we're not targeting protein synthesis. And the other thing that we show is if we add a pyrase to the cell-free plate screen, even if we add the compound prior to puro release, we do not get the drug effect, meaning even though it's a very early mechanism, in order for the drug effect to be manifest, it needs the later energy-dependent steps of the capsid assembly pathway to be manifest. So just to summarize, taken together, the studies that I've talked about advance an alternative view for capsid assembly, specifically a host-catalyzed pathway, a different paradigm for drug discovery, right, whole pathway screening without knowledge of the target, and suggest evidence for the existence of labile assembly machines that can be rendered accessible as next-generation drug targets through the approach that I've talked about here. And this work was funded by the NIH and the Northeast Biodefense Consortium, and I'd like to thank our collaborators at the CDC for help with the infectious virus studies and the whole team at Prosetta.
This is a very interesting talk. Have you ever, so presumably you're targeting host-specific factors. Have you ever tried to generate drug-resistant virus from any of your compounds that inhibit?
No, we haven't, not yet. We're still in pretty early stage.
Early stage, yeah.
Have you tried the same compounds in other virus, assembly? Because you're mentioning this one is also in HIV and other.
Yeah, so we've established similar screens for a number of different viruses, and have screened a lot of the same compounds on all the screens. And we found some overlap in activity, but in initial hits. But usually when we do the SAR, they separate out. Yeah.
Yeah, I feel the capsid name is confusing because normally we say capsid is outside of virus. I think you mean the nuclear capsid, right?
Yes,
complex,
yes.
So you haven't tested any of the drug in even cell culture system to see if the drug inhibit the viral replication in cell culture.
We have tested in cell culture. Yeah, and we've found that it does inhibit.
The in vivo, have you got a chance to do the animal study?
We've not done animal studies yet, but we hope to in the very near future.